So, very good afternoon to all of you. So, welcome to all the participants and the speaker to this afternoon session of 21 days training programs. Today, we have with us Dr. Neeraj Kumar Singh. He's working presently as a scientist at College of Animal Biotechnology, Guru Angad Dev Veterinary and Animal Science University, Gadwasul, Ludhiana. Dr. Neeraj was a graduate from Maharashtra Animal and Fisheries Sciences University, Nagpur, and did his master's and doctorate in animal biotechnology from Indian Veterinary Research Institute. He has published He has published 25 papers in peer-reviewed journals, review papers for book chapters, 3 popular articles, and 5 practical manuals. His area of work are molecular biology, vaccine development, diagnostic, and viral host interaction. So he's today talking about the basic principle and concept of biotechnology that are mostly useful in plant as well as animal sciences, and particular in reference to animal sciences. So I welcome Dr. Neeraj from the organizing committee. Over to you, sir.
Thank you, Dr. Pragya. Thank you very much. I am Dr. Neeraj Kumar Singh. I am working as scientist in College of Animal Biotechnology, Gadwasu, Ludhiana. My topic of today presentation is biotechnological interventions in animal sciences. So first of all, what is biotechnology? Biotechnology is the use of biology. Biotechnology is the use of biology to develop new products, methods, and any organism which with the intention to improve human health as well as the society. So biotechnology is not new science; it is very ancient science. From ancient times, people has been using the practice of biotechnology for their day-to-day life. The best example of the biotechnology used in ancient time was there is use of cheese preparation, curd preparation, alcohol preparation. These are the some examples which indicate that people was aware about the biotechnology. How the biotechnology intervene the life is the people was aware, and with the help of biotechnology, people was benefited. In pre-20th century, there is big development happened there in the field of biotechnology. The most importantly, in 1519, Johnson invented the microscope. With the help of microscope, people can see the organism which cannot be visible at all by the naked eye. After these inventions, in 1663, Robert Hooke discovered cells. Subsequently, Leeuwenhoek discovered the bacteria and protozoa. And in 1797, the Jenner, who is considered as the vaccines, who is considered as the father of vaccinology, has first time given the brief about what is vaccines, how vaccines work. Subsequently, in 1855, Pasteur, he was the great biologist who given several vaccines during those times, including the vaccines against the rabies. Pasteur also given the concept of pasteurizations, which we are till to date we are using the concept of pasteurization for milk. That was given by the Pasteur. In 20th century, before the 1950s, some development was there in the field of biotechnology was the term genetics given in the 1906. In 1915, bacteriophage was discovered. In 1990, the Carl Erke, he has given the term biotechnology. Actually, Carl Erke considered as the father of biotechnology. He was one engineer. He shown how the waste material can be converted into very useful product. So he used the waste material, sewage waste materials and other household waste material in the pig farm. And after pig farming, he had extracted so many biologically important products, including collagens and other things are there. So he saw how biology can be intervened by the biotechnology to produce very useful product. Subsequently, in 1928, first antibiotic penicillin was discovered by Fleming, Alexander Fleming, and later on the molecular biology terms come in existence, and the genetic engineering and other terms comes in existence. After 1950s, this big development happened in the field of biotechnology. In 1954, the techniques for the cell culture was first developed there and first are in used. Subsequently, in 1956, the fermentation process was more commercialized and more perfectly used there. In 1977, first genetically engineered bacteria were developed to produce useful human growth proteins. In 1978, side diagram mutagenesis was discovered. In 1979, first monoclonal antibody and hybridoma technology was produced there. In 1981, genetically engineered plant were produced. In 1982, genetically engineered bacteria was produced, which was used for the production of human insulin. This human insulin nowadays known as humulin. There is very big breakthrough happened there in 2000s when first draft of human genome project was prepared. In 2001, this draft was published in the Nature and Science journals. So, how, in which field biotechnology play roles? Biotechnology play important role in the field of medicines, agriculture, animal husbandry, food processing, and environment science. Out of these many science, I am here only to focus on the how biotechnology play important role in the field of animal husbandry. So actually, biotechnology play important role in the animal husbandry by three different ways. First way is by improving the animal productions. Second, by improving the animal health, and thirdly, by improving the production of some useful product for the human being or for the other purposes by animal sciences. So how biotechnology play roles for improving the animal productions? Actually, biotechnology play in several ways to improve the animal production. Firstly, to identify the superior animals at the early. Normally, suppose if we want to characterize any animals for some specific purpose, like milk yield, if we are thinking, okay, these animals will, how this, what is the this, how much this animals is milk yielder. So for that, we have to wait for the at least up to the age of 4 or 5 years of a cattle, because after first and second calving only we can find, okay, this animal is high yielder or this animal is low yielder. But with the help of, with the intervention of biotechnology, we can identify day one old calf that whether this will be in future high yielder or this will be low yielder. So biotechnological intervention can use for the predictions of the animal performance in the futures. Secondly, biotechnology can be used for the increased rate of genetic improvements. Traditionally, how genetic improvement are achieved? Normally, for normal breeding process, normal breeding process are too long, too lengthy. So it is very lengthy for getting one generation's calf and subsequently to become mature. At least 7 to 8 years required there. But with the help of biotechnological interventions, we can increase the rate of genetic improvement, especially in field of the cattle. In India, you know, there the religious issues are there. We cannot go for the culling of the cattle by slaughter methods. So with the help of biotechnological intervention, we can bypass the rules of the selections of the animals through the slaughter or through the culling. So biotechnology also play important roles for increasing the rate of genetic improvement. Thirdly, to detect abnormality. As already I have told, that some abnormality which are related with the age, that abnormality can be only detect only attending after some age. But with the help of biotechnology, we can identify the genetic abnormality at the very early age. Like we can go for amino synthesis through the amniocentesis, we can collect some fetal cell. By molecular technique, we can identify whether the calf will generate produced by this fetus will have some genetic abnormality or not. So in uterus only, we can identify the genetic abnormality. Fourthly, improve understanding of mechanism of genetic control. So biotechnology also play important role to study about the genetic controls on the different phenotype. Fifthly, to determine the parentage. So as you know, the parenting testing are there nowadays. Previously, it was commonly in use for the human being, for the criminal cases. But nowadays, parentage testing can be also performed for the human being with the help of biotechnology. Fifthly, transgenesis. So with the help of transgenesis, we can improve the animal productions, which I will discuss later. Then animal nutrition. Biotechnology also play important role to improve the nutritional quality of the food of given animals, or to improve the digestion mechanism of the given animals. So also I will discuss in the later slide. Now, how, as I have told, to genetic improvement, how the genetic improvement can be achieved. For genetic improvement, first of all, we have to identify what are variations among the animals, genetic variations among the animals. Normally, in a populations, 99.95% nucleotide sequence are similar. So suppose 1,000 animal populations are there. Among 1,000 animal populations, all animal have 99.95% similar nucleotide sequence. Only 0.05% variations of nucleotide happen there in the genomes. So these variations have to identify what are different variations happen there in the genomes. So these variations can be correlated with the phenotype. Normally, two individuals are not carbon copy to each other. This variation is due to the genetic variation, and genetic variation is very less. Very less means only less than 0.05% variation there, responsible for all phenotypic variations. These genetic variations are known as single nucleotide polymorphism. So we have to firstly identify the variation among the genetic variation among the individual, then subsequently have to develop SNP, single nucleotide polymorphism, which are related with the phenotypic variations. Then we have to relate these SNP differences with production differences. Suppose two animals are there, one animal are very high yielder, another is low yielder. So after complete genetic study, we have to correlate what are the different genetic variations are there. So what are the different SNPs are there, single nucleotide polymorphisms are there. So these genes, we have to correlate whether these SNPs are related with the milk yield or not. So we can relate the SNPs with the production differences. After correlating SNPs with the production's performance, we can select genetically improve animals for future breeding purpose. So only genetic material of high genetic merit animals can be passed from one generation to next generation. So these are the way by which we can implement the biotechnology for the genetic improvement. So first of all, again I am repeating, for genetic improvement, what we have to do, we have to characterize what are the different genetic variations among the populations. These genetic variations are known as SNPs. We have to correlate SNPs with the production differences. After making relations between the SNPs with the production differences, we can select the animals with high genetic makeup, which will be in future produce high performance. Subsequently, this selected animal can be used for the breeding purpose. So only high genetic materials will be used for the breeding, and low quality genetic material automatically will be culled. So before the going for the this one genetic this one, what is genetics? What is genomics? What is genomics? As you know, gene is the oligonucleotide sequences which is responsible for a polypeptide sequence. But genome represent all genetic materials of the cells. So include all genes. Normally in human being, approximately 50,000 genes are noticed, and its genome size is 3.2 billion. So genome is nothing but it is the complete genetic material of a cells. For human being, the complete genetic materials of a given cells having 3.2 billion bases. So it is very big gene. That genome materials is very big. To characterize genetic materials, complete genome is not easy. As I have told, the first genome project draft was completed in 2000s. That work was carried out for 6 years by 20,000 scientists. So for one genome, or one human genome, complete sequencing for complete characterizations, 6 year time was taken with the help of 20,000 scientists. So it is very, very tough and tedious task. But with the advancement of biotechnology, nowadays, one scientist within 6 days can characterize complete genomes of a given organism. So biotechnology, with the help of biotechnology, the genomic study become very fast. Nowadays, the complete genome sequence of the poultry and livestock are available on NCBI data bank. So we can easily identify the what are the genetic material or what is the genetic composition of different animals. Now this genetic material or genomes can be further narrowed down into the functional genomics. So all genomic materials, all genomes, as I have told that for human beings, 3.2 billion base pairs are there. Out of 3.2 billion base pair, less than 2%, so less than 6.06 billion nucleotide actual contribute. 98% and above genome is non-functional, has no function, only less than 2%. So after study of the complete genome, we have to only target the part of genome which is actual contributed for the phenotype. So this process is known as functional genomics. So this science is known as functional genomics. So how genomic can be used? So the genomic can be used for identify the sequence associated with the disease resistant and production trait. So after getting the knowledge of the complete knowledge of the genome, we can identify whether animal will be have high performance capacity or animal have high resistant against the disease. We can identify only after the genome with the help of biotechnology. And secondly, with help of the genomic knowledge, we can select the animals even before the birth. Normally, selection of animal, normal practices, what is there in the farm practice are there in day-to-day practice. After performance, after milking, after first calving or second calving, after first complete milking and some second complete lactations, we can characterize, okay, this is low elder or this is a high elder. But with help of the genomic complete, after the complete genomic informations, we can select, we can evaluate the animals even before the birth. Even the birth animals are there in the mother womb, we can evaluate whether animal will be the high producers or low producers. And only high producers or high genetic material animals will be used for the further breeding purpose. So now genetic marker. So marker, as you know, what is marker? Marker is some important significant features. Suppose for human being, someone has been a cut mark on the chin. That is marker for that animal, that in the human individual. Second one having someone cut below the eye. So that is marker for that second individual. Likewise, for human animal beings, genetic markers are there. Genetic marker is nothing but it is SNPs or some oligonucleotide sequence variations which related with the performance of a given animal. Marker can not be required by part of gene. So I am not here to discuss what is gene. So marker, as I told, marker, there are three kind of markers: phenotypic marker, biochemical marker, and molecular marker. What is phenotypic marker? Like human being. Someone is whitish color. Someone is dark color. Someone is whitish color. So these are the, for Indians, Indian are whitish color. Negroid are blackish color. European are whitish color. So for European, what is marker? If someone is a European, that means they have whitish color, skin coloration. हाँ शिखा मैं बाद में बात करता हूँ क्लास ऑनलाइन क्लास so there are three kind of marker as I have told. One is phenotypic marker that can be determined after the simple physical appearance by simple vision. We can characterize. This is the marker for this, for European races animals. European races human beings, whitish color. Indians races, whitish color. Negroid, blackish color. Chinese, small in size, height. Second one is biochemical marker. Some important biochemicals which is associated with some specific individuals only. That is biochemical markers. Third is molecular marker. I have told SNPs. SNPs are also a molecular marker. Single nucleotide polymorphism is a molecular marker. The molecular marker can be either single locus marker like SNPs or microsatellite marker, multi-locus marker. These multi-locus markers are commonly used for the DNA fingerprinting. You know what is DNA fingerprinting? DNA fingerprinting, the characterization of individual on the basis of their genetic makeup. So DNA fingerprinting are commonly used for the criminal cases or forensic cases or for the animal selections. So this is brief about the molecular markers. Now, how the molecular marker, how the SNPs are used for the animal improvement. First of all, we have to see the phenotype and correlate this phenotype with genotype. What are the genetic variations? So first we have to see the phenotype and correlates with the variation in the genetic material. After the correlations, we have to identify some markers which is related with that important phenotype. This after selecting markers, we have to go for the marker assisted selection. Means that suppose at one locus there is 4 possibility of different nucleotide A, T, G, C. Suppose A or T nucleotide is there, that means that is high yielder. If it is G and C, it is low yielder. Then we have to go for the genetic profiling of the different individuals for that locus, for that positions, and the animals which having only A and T had to select, and C and G has to reject. So this is known as marker assisted selections. First, we have to identify the markers. Second, the markers which is related with the high yield has to characterize, and thirdly, the animals which has that specific markers related with the high yield has to select. That selected animal has to use for the further propagation. So the progeny will have the high yielder. So this all process is known as marker assisted selections, MAS. Very important. This marker assisted selection not only applicable for the animal beings, but it is also applicable for the plant or agricultural sciences. Now, apart from genetic improvements, suppose the genetic improvement normal are there, that is time consuming. But nowadays, there are several assisted reproductive technologies or ART. With the help of assisted reproductive technology, we can lower down or we can cut down the animal reproduction time or animal selection process. We can cut down with the help of animal ART, with the help of animal assisted reproductive technology, we can lower down the time required for the animal genetic improvement. These animal ART techniques are artificial inseminations, OPU, IVF, and somatic cell nuclear transfer. Many of you are from the field. They know what is artificial inseminations. Artificial insemination is the process in which the semen of high yielder animals, genetically proven bulls, has to use for the breeding purpose. So, in artificial insemination, the high genetic materials of male only propagate in the next generations. In this artificial insemination, we cannot go for the selection of high genetic material of females. So artificial insemination is important techniques that can be used for the genetic improvement. In India, in 1970s, the artificial insemination was started there. Due to artificial inseminations, the milk yield increased drastically. Now India is highest milk yielder because this is due to artificial inseminations. But the disadvantage of artificial insemination is that we have lost our own germ plasm. Now if you around you, you can rarely find a pure breed Indian breed cattle. Here I was someday before I was in the field, I was even not able to get single pure bred Sahiwal cattle or pure bred Sahiwal, no, sorry, Gir cattle. Almost all animals have some contaminations of genetic material of exotic breed due to artificial insemination. Due to artificial insemination, of course, we have improved our milk productivity, but at the same time, we have lost our own genetic materials. So this is advantage and disadvantage of the artificial insemination. Second is OPU IVF. As you know, now Government of India has launched one important program in many centers, especially in the ICR institutes, Animal Science Institute, and different universities like Rashtriya Gokul Mission, RGM. I think many of you know what is RGM. Rashtriya Gokul Mission is there, where this OPU and IVF practice are in use. Here in OPU IVF practice, not only the highly high genetic materials of male, but also high quality genetic material from female can be propagated. In artificial inseminations, only genetic materials of high producers male propagate. But here in OPU IVF, the genetic material of high yielder male as well as female can be propagated. So what are the steps are there? So first of all, OPU. For OPU, the high yielder animal has to select, female animal have to select, go for hormonal super evolution. After hormonal super evolution, have to collect the ovum and culture there in the in vitro condition. After culturing, the ovum has to fertilize by the semen from the high yielder bulls. Now here the semen as well as ovum are from the high yielder animals, high performance animals. So here the not only the male but also females which has high production capacity can be propagated. After fertilization has to subsequent culture for 7 days, then blastocyst will be developed. In blastocyst, either can be stored for the future purpose or can be directly transplanted into low yielder animal. Here, this is low yielder animal, so this is act as an incubator. The progeny produced after this progeny won't have the genetic material of the mother who has given birth, but it has the genetic material of the main mother, biological mothers, from which the ovum has come out. So this is surrogate mother. Surrogate mother is low yielder, main mother is high yielder. Here in OPU IVF technique, we can propagate genetic materials of high yielder male as well as females. So these are the advantage and disadvantage of OPU IVF techniques. Now SCNT, somatic cell nuclear transfer. I think all of you know cloning. The first cloning was developed in Dolly. You know, everyone know, I think. But who is Dolly? Who was Dolly? Dolly was the first cloned ship in 1997. The ship were produced there. That was Dolly. Now, if you visit Europe, you can see on simple one clinic, people are doing the cloning. But for India, it is still very rare practice there. Cloning is very rare practice in India. In India, in my knowledge, in my best knowledge, only 2 animal science institutes where the cloning are going on. Cloning can be performed by various techniques. The most common technique which is applicable in India for cloning purpose are somatic cell nuclear transfer. The institute where the clonings are going on there, that is NDRI, National Dairy Research Institute, Karnal, and another is CIRB, Central Research Institute on Buffalo. So these 2 institutes are working on cloning. So cloning is nothing but it is the process in which a enucleated ovum. So how we have to do? First, we have to collect the ovum from one females. Then we have to remove the genetic materials, all genetic materials. That is nucleus. This is haploid nucleus. Now the ovum is there. This is completely enucleated. It is not having any nucleus. Now either we have to take one nucleus from one diploid cell and transfer, or directly fuse one diploid cells with ovum. After fusion, the cell will produce that has some feature of ovum as well as chromosome of diploid cells. This fused cells or this nuclear transfer cells has to given some chemical or electrochemical stimulus. After giving this electrochemical stimulus, these cells start to multiply and to develop the blastocyst and blastula. Subsequently, this will be develop fetus and produce the cloned animal. So this cloned animal has all feature of the animals from which the diploid nucleus has transferred. So this is now techniques, very rare techniques in India, but for Europe and US, it is most common technique for the animal cloning. People are doing there the pet cloning. Someone having very much love for the dear dog like pets, dear pets like dog. If the dog is going to die, then they have to go for cloning. So they have carbon copy of their pets. They will get the carbon copy of their pets. In India, this SCNT technique are very rare in use. This is only 2 institutes there doing the SCNT, single somatic cell nuclear transfer technique. One institute is NDRI Karnal, another institute is CIRB, in my best knowledge. Other institute, other than ICR institute, wherever it's working going on, I am not aware about that. Now, other important aspect is for the animal improvement with the help of biotechnology in semen sexing. What is semen sexing? As you know, for normal breeding process, there equal chance of getting male as well as female. 50% male, 50% female. For cattle, male are of very less use. Nowadays, it's no use, almost no use. Previously, the male was used for the draught purpose, for plowing purpose. But now, plowing is not practiced by the help of ox. So, male are of almost no use. So, all farmers are willing to get their female calf only. With the help of biotechnological interventions, we can segregate the sperm for the male as well as for female. So after sperm sexing, we can separate the sperm which can produce only female calf. So semen sexing can be achieved by MAX. This picture represents a MAX, magnetic assisted cell sorting. Other techniques are also there, like flow cytometer and density gradient. This is not given there. I'm just making aware of what are the techniques can be used, what are the practice in use for the animal improvement. So one technique is there, semen technique, sexing. So semen, with the help of semen sexing, we can get progeny of one sex only. So if we get the 100% or 90% female, so no male will be there, no male calf will be there. So we can increase the productivity. Another is animal genome editing. So what is genome editing? What is editing? Computer science or normal editing means to make some changes. So here, what is there? How can we genetic material can be done? With the help of recombinant DNA technology, we can make the genetic changes there. Editing. There are different processes of the genome editing. This is their embryonic stem cells are there. In the embryonic stem cells, we can make some changes there. After getting changes inside the cells, we have to transplant these cells into the blastocyst, and this blastocyst has to use for the pregnancy purpose. Second, another approach are there. Zygote has to edited, or third approach is the SCNT technique. So different various ways are there to use for the genomic editing purpose. Now the questions: what is the use of the genomic editing? The most important example, 2-3 example I am telling you. All of you know about the A1, A2 milk. I think everyone knowing what is A1 milk, what is A2 milk. It is normal perception is that A2 milk is better than A1 milk. A1 milk having some harmful effect. It is not scientifically proven. It is people are thinking. So normally exotic breed are producing A1 milk, our indigenous cattle producing A2 milk. It is due to single nucleotide variations, only one nucleotide variations. If we can able to make change of that nucleotide in exotic breed, then we can get A2 milk from exotic breed also. Like HF or Jersey is there. They are producing A1 milk. Our indigenous breed, cattle like Sahiwal and Gir, producing A2 milk. This is due to single nucleotide changes. One nucleotide is there, variation there between A1 and A2 milk production animals. By any mean, if we can change one nucleotide, then from exotic milk breed, we can get A2 milk. So with the help of this genetic manipulation, that people are doing that. This has already done so many work on there to producing A2 milk from exotic breed also. The second example is myostatin knockout bull. As you see there, this photo of bull, this is not normal bull. Here the muscle mass is double of the normal bull. So normally, for normal physiology, one important proteins, that is myostatin, responsible for proper development of muscle mass. If by any mean, if you remove this myostatin proteins, then the animal will produce, which will have uncontrolled very high muscle mass. This can achieve by removing the gene of myostatin. So myostatin knockout can be used for the production of animals with high muscle mass. For the animals used for the meat purpose nowadays are produced by myostatin knockout process. In India, two institutes again, NDRI Karnal and CRE, very shortly, within 2 months, they are going to, their 6 calf are going to be, 6 buffalo calf are going to produce, which will have myostatin knockout. So that buffalo will have double of muscles than in compared to normal buffalo. This is the picture represent. Now people are working on the myostatin knockout for the goat, myostatin knockout for the cattle. Already I have told in India already. People very shortly we will get. Then cattle Europe. Then this one pig. Then this is sheep. So people are working on the myostatin knockout. So that animal will produce high meat. So we can get the all requirement of meat by with the intervention of biotechnology in the animal sciences. Now, how biotechnology play important role for animal production, for animal nutrition? Firstly, With the help of biotechnology intervention, we can produce a crop which has high nutritional quality. Suppose we are using some crop of maize which has high content of carbohydrate. With the help of biotechnological interventions, we can increase protein content of the maize. So that genetically modified maize can be used for animal feeding. And if animal will get the high nutritional food, that will produce high quality, that will produce high amount of product. Second in way by which the animal nutrition can be improved, that is by the manipulation of the ruminal microbes. In human, prokaryotics are there, yeast are there, protozoa are there. In prokaryotic, gram-positive bacteria, gram-negative bacteria, both kind of bacteria are there. Gram-positive bacteria are normally nuisance bacteria, and gram-negative bacteria that are favorable bacteria. So, with the help of biotechnological intervention, we can manipulate the microbial content of the human. And after this microbial manipulation, the animal production can be improved. So, these are the way by which animal biotechnology can use for the improvement of the animal production. Now, second aspect of animal biotechnology for improvement of animal health. Animal health can be improved by either prevention of disease or by diagnosis of disease or by therapeutics. Here I am discussing about two aspects, either vaccines or diagnostics. So biotechnology has important role for the vaccine development. Till today there are 4 different generations of vaccines are there. The first generation vaccines are either live attenuated, whole pathogen, or inactivated, killed pathogens. This is the first generation vaccines. The Pasteur developed this first generation vaccine, inactivated vaccines. Okay. Subsequently, the advancement of vaccinology was happened there, and second generation, third generation, fourth generation vaccines were developed. The second generation vaccines having subunit vaccines, toxoid vaccine, tetanus toxoid (TT) are the second generation subunit vaccines, conjugated vaccines. Third generation vaccine, nucleic acid vaccines, DNA vaccines. Fourth generation vaccines, recombinant vaccines. The best example of the recombinant vaccines are Covishield. What is Covishield? Covishield, in this Covishield, a adenoviral vectors used for the carry a gene of SARS coronavirus or COVID virus. I will discuss this one in later slide. I will discuss about the Covishield as well as the Covaxins. This is the Covishield having recombinant viral vaccine, and Covaxin is inactivated or killed vaccines. Then genetically manipulated vaccines like MDV, VHV, gene deletion vaccines, marker vaccine. And another is chimeric vaccine. What is chimera? Chimera means the individual with 2 different features. The individual with the feature with two different individuals, 2 different species. Like in Hinduism, nursing. You have heard Lord Nursing. Nursing was having head of lion and rest of body of human. So likewise, here we have to produce the microbes which has some portion of one pathogen and some portion of another pathogens. So this will provide protection about these both organisms, like Brucella and BHV vaccine. Brucella and BHV both are related with the animal reproductions. So this chimeric virus has some part of BHV virus, bovine herpes virus, and some part of Brucella bacteria. This chimeric organism can provide protection against both BHV as well as Brucella. So this is vaccines. Already I have told earlier, so I will discuss about the Covishield and Covaxins. Covaxin is the first generation vaccines. Here in Covaxins, the coronavirus has to inactivated. After inactivations, after killing of this coronavirus, it is used as a vaccine, and this vaccine trigger the immune response and provide the immunity against the COVID virus. Second way of the vaccines in the use in India is Covishield. Covishield is in COVID, you see here, this is the picture of coronavirus. Coronavirus is having a spike proteins. The gene which is responsible for a spike protein has to incorporated into the complete genome of adenoviral vectors. So this adenoviral vector, recombinant adenoviral vectors, infect after injections, infect the cells, and infected cell will produce the spike proteins of coronavirus and provide protections against the COVID. So this is one approach by which biotechnological intervention can provide immunity to the animals. Second is diagnosis. You know, prevention is better than cure. So first, if somewhere outbreak is there, you know, everyone has passed through this COVID pandemic, the isolation, quarantine. Why this is in practice? This is in practice to prevent further spread. So for further spread prevention, you have to diagnose whether animal is positive or not, or individual is positive or not. So traditionally, diagnosis are there, simple by physical appearance. Subsequently, the diagnosis was improved and this one, serological diagnosis like ELISA, AGID, AGIT, SNTs was in practice. Nowadays, molecular diagnostic was in very much useful. The molecular diagnostics are PCR-based diagnosis, fluorescence in situ hybridization-based diagnosis, nanotechnology-based diagnosis, biological chip is there, PNAs are there, proteomic technology are there, electrochemical detections are there. When I was in masters, during that time, I was thinking that PCR is a very good technique, but it cannot be implementable because it's cost as well as technical expertise required. But this corona pandemic has shown no, any technique which has very high cost and require very high skill can be implementable at the mass level. The best example is the real-time PCR-based diagnosis. Everyone knows, I think everyone has passed through the qPCR-based diagnosis for the COVID. So this is the pictorial representation how qPCR can be used for the diagnosis purpose. Now, third way of biotechnological interventions for animal science is to produce some useful product from animal sciences. The first one is the hybridoma technique. Hybridoma means what? Hybrid and oma. These two terms. Hybrid means two, fusion of two, and oma means out of two, one is cancerous. So hybridoma is technique in which antibody producing cells get fused with cancerous cells. This antibody producing cell having feature of producing antibody, but it has very short lifespan. While cancerous cells, it won't produce, it is not producing any antibody, but it has infinite lifespan. After fusing these two cells, the cell producing, that is known as hybridoma. This having feature of these two cells. Normally, these cells having very limited lifespan and producing antibody. This hybridoma cell having get one feature from these cells. What is the feature? That is antibody producing feature. And another feature, that is infinite lifespan, got from tumor cell. So hybridoma cells having feature of antibody productions and it has infinite lifespan. So antibody, first hybridoma technology was developed in 1975 by Kohler and Milson, and they got the Nobel Prize in the medicine in 1984. So hybridoma technology used for the production of some useful product that can be used for the animal science, human health, as well as plant science. Then transgenic animals. These are some picture of the transgenic animals. These are fancy animals. You see, this is not normal color animals. This is having some green fluorescence color. By use of the genetic manipulations, transgenic animals or genetically modified organism has produced. In this picture, the transgenic mice and transgenic rabbit is here represented. This transgenic animals has green fluorescence proteins. So this green fluorescence proteins give fluorescence color in UV light. Another is transgenic sheep, transgenic pig. I will discuss in later slide. Another is transgenic cow and fish. So I will discuss in later about the transgenic pig and transgenic sheep, how it is useful for the human, for for a human purpose. So organ transplantations. Many one have known about the organ transplantations. Organ transplantation means transplant the on animal, transplant organ from one organism to another organism. Okay, so transplantation either may be autograft, maybe allograft, or maybe genograft. Autograft was one organ from one part of same individual transplanted to other part of that individuals. Like some skin burn is there. Someone has a skin burn on the face. For that case, a small skin patches from the thigh region has to taken out, has to grown in the culture, and has to implanted on the face region. This is known as autograft. In autograft, there is no graft rejections. But allograft, what is allograft? Organ of one individual, suppose kidney of one X individual, has to transplant it into the Y individual. The important things is there. This has graft rejections. Means the persons who has received the organ from another individual had tendency, their immune system had tendency to reject, to kill the incoming organs. For prevention of that, there is immunosuppression therapy is there. Third approach is there, genograft, means organ of one species has to transfer into the another species, like human heart valve transplantation. That heart valve from the pig has to transplanted into the human beings. This pig is known as humanized pig. Humanized pig. What is humanized pig? So in humanized pig, the MSC, major histocompatibility complex of pig, has to removed, and MSC, major histocompatibility complex of human beings, has to incorporate. So this pig do not has MSC of pig, but it has MSC of human being. What is MSC? Already I have told, graft rejections. Graft rejection means organ has to rejected by individuals due to immunological rejections. This immunological rejection happened due to MSC compatibility. Only the MSC is the important factor which is determining for the graft rejection. With help of this genetic manipulation of incorporation of MSC of human being and removal of pig own MSC, this pig organ can be transplanted into the human being. This transplantation is known as genotransplantation. Another way for the organ culture is there. First, we have to take some cells of the human beings, have to implant it into blastocyst. This pig will produce, this pig will have some organs which have the human cells, then that cells can be transplanted. But this is very rare technique. This techniques, genotransplantation, humanized pig are nowadays in use in Europe as well as the America. Now biofarming. Biofarming is what? Use of biological system for production of some specific product, especially human, especially animal being. Here animal beings is there. This is normal sheep is there. Normal sheep, what is it is doing? Normal sheep producing milk. It having normal casein, proteins, and other factors are there. But with help of genetic manipulations, we have to incorporate some desired gene or a specific protein into the zygote of sheep. After incorporation of desired gene, the full animal will produce. Inside this whole animals, this gene, our desired gene which has incorporated, has to express there. Like most commonly in the use there, production of antithrombin proteins in the milk. This is genetically modified. Sheep is there. This produce milk which has antithrombin proteins. Heart attack or hemorrhage, cerebral hemorrhage. The two important nowadays. These are the two important factors which is responsible for death. This death happened due to the clotting. Blood clotting is there inside the heart, or blood clotting inside the brain. That is the major important factor now is for the death, human death. Now, what happened there? For dissolving the blood clot, we need thrombin protein. We need thrombin proteins, antithrombin proteins. So antithrombin protein, how can we can get? We have to get the antithrombin genes is there, has to incorporate into the ship zygote. This ship zygote will implanted into the mother. Subsequently, the newborn ship will be produced. This newborn ship has genetic material of antithrombin genes. This ship will mature, will produce milk. This milk has antithrombin proteins. So this protein, consumption of these proteins can be used for the treatment of heart attack as well as the cerebral hemorrhage. So in conclusion, biotechnology, whatever I have discussed till today, this is very small part. This is less than 0.1% part of biotechnological interventions in animal science. Still, there 99% part are still uncovered. That one biotechnologist can only understand. I have prepared this slide by considering that someone is from the field veterinarian, someone from the SMS, someone from the unrelated field like veterinary, veterinary anatomy, veterinary LPT technology, LPT. So we have prepared this generalized slide. 99.9% part still has uncovered. These are the some in conclusion set. In conclusion, I can say the biotechnology has its application in animal science in different way. These are the some picture representations. So this is all about the biotechnological intervention. Brief about the this is all brief about the sorry sorry. This is brief about the biotechnological interventions in animal science. You know, nowadays almost every field of science, including agriculture science, fishery science, whatever field you go there is separate department for biotechnology. But it is very sorry to say, until now, there is no separate department as per BCI in the veterinary course curriculum. So thank you. So now your questions are most welcome. Hello.
Thank you very much, sir. It's a very elaborative and very great insight of biotechnology in animal sciences and related field. I think the audience will definitely get benefited by it. And I would like to add that I forgot to mention that sir has worked with DBT in many projects, as well as he is appreciated by Punjab government because he was involved in detection of corona. He has worked very hard during that COVID testing lab, single lab at Punjab in Patiala, I think. So, sir was very renowned scientist of biotechnology. So, if there is any question related to biotechnology or some related aspect, because I told to sir that it should not be means audience are of multidisciplinary, so they should get a grasp of this subject. So I would like to thanks sir for giving his valuable lecture on this topic, important topic, because we have to get a proper knowledge of everything. So if there is any question, you can write in the chat box. Sir, there is one question in the
They can
chat
ask.
box. They can ask, and chat box there are questions also.
Okay. Please questions.